Hello, my name is Michael Johnson, and I'm the CEO and one of the co-founders of Visikol. Today, I'll be talking about the Hurel portfolio of microliver products and services. Now, before I dive into talking more about the Hurel products and services, I wanted to give you a little bit of background about Visikol as a company. Visikol is focused on accelerating the drug discovery and development process by providing pharmaceutical and biotech companies with advanced cell culture models and assay services, as well as advanced tissue imaging services. These are the two pillars of our business, and we've worked with 18 of the top 20 pharmaceutical companies in helping them accelerate their drug discovery and development processes, as well as dozens of other small and mid-sized pharmaceutical and biotech companies. Now, the focus of today's presentation is on advanced cell culture. And advanced cell culture, the objective is to try and bridge the gap between in vitro studies and in vivo studies. Ideally, what we would like to do is have in vitro studies mimic what happens within the body in an inexpensive and high throughput format so that we can predict what's going to happen very early on in the drug discovery process. But unfortunately, this isn't always the case. So we're constantly trying to better mimic the body in a dish and develop complex models that can accomplish that task. We as a company are focused on liver cell culture models and assays, as well as oncology models and assays. This tends to be about 80 to 90% of the work that we do. In the liver space, we have several models, which I'll talk about, that have been developed and validated for various applications. And in the oncology space, we also have several models that tend to be focused on immuno-oncology applications, such as the T-cell infiltration assay that I'm showing here. We're quantifying how T-cells are infiltrating into a spheroid model of cancer. And then lastly, while we focus a lot on oncology and liver cell culture, we as a company are model agnostic. We've developed all sorts of models for custom applications and disease states. And as a company, have worked with everything from transwells to ex vivo tissue slices, and really can focus on developing custom assays and models for projects based upon a customer's specific requirements, and are very open to doing that and working hand in hand with our clients. But the focus today will be on liver cell culture assays and models. A really important thing to consider is that there is not a one-size-fits-all in vitro model. 3D cell culture, bioprinted models, tissue slice models, they're not going to replace all the other types of models. Every in vitro model has its place because when we look at a model, it's a balance between throughput, price, and relevancy. A model can be extremely relevant, such as a tissue slice model, but the price can be quite high and the throughput can be quite low. So depending upon your research question, there's a specific model that meets those different criteria. And this will depend on what question you're specifically asking, what your budget is, what your throughput requirement is, how many compounds you're looking at, and also what types of in vivo recapitulation do you need to actually answer your research question. If you can get by with a very simple model that's at low cost, why not use that model? So we're always focused on this as a company in trying to provide models that meet our clients' specific criteria for their assays. As a company, we have a broad spectrum of models spanning from 2D cell culture all the way to ex vivo tissue slices of human, rodent, feline, and also canine for use in the liver space. Prior to acquiring Hurel in May of 2021, we were very focused on spheroids, organoids, and tissue slice models, all 3D or tissue slice models that are quite complex, challenging to characterize, and useful for various applications, but not all applications. These models tend to have metabolic stability for only a few days to a few weeks, and phenotypic stability is not as strong as it is in some other models. And this is one of the main reasons why we wanted to acquire Hurel. Hurel, as a company, their services and products were based upon 10 different species of primary hepatocyte co-culture 2D models. They were extremely stable and stable typically and also metabolically for long periods of time. And thus, by adding this portfolio of models to our portfolio, we're now able to provide our clients with the broadest breadth of liver cell culture models in the entire marketplace. From 2D to spheroids to organoids to tissue slices, we have all these different models within our portfolio that we can pull into DMPK studies, looking at non-alcoholic steatohepatitis, looking at fibrosis, looking at immune cell infiltration into liver tissues, and also traditional drug-induced liver injury. So we have a very broad portfolio of liver models as a company, and the focus of today's presentation, though, is on the Hurel microliver models, which I'll get to in a second. But what I'm showing here is not every model answers every research question, and it really depends on what that research question is that we're going after before we pick which model we're going to use, because there's not a one-size-fits-all solution. The Hurel microlivers, which are now part of the Visicol portfolio of models and assays, are a 2D liver co-culture system. The process by which we culture these systems is patented and proprietary, but it allows us to keep primary hepatocytes in culture and metabolically and phenotypically stable for over 4 weeks, up to 2 months of time, which is something we just don't see with more complex spheroid models and something we don't see in traditional 2D cell culture. So it's a very unique model, but it's also very well validated. We have over 100 publications using this model for various applications. We sell it as both a product and also leverage it in our services, which I'll get to in a moment. But as a product, it is shipped live, and it can be shipped in 12, 24, 48, 96, and 384 well plate formats ready for use in your assays. And because it's a standard plate format, it's compatible standard automation and analytical equipment. Any imager, any other type of equipment that you've used in your lab for plates is compatible with this system. So we don't have any special fluidic devices. We don't need any special equipment. This is a standard plate format, and it's used by most major pharmaceutical companies for various applications. Now, as I mentioned, this has been in various publications for lots of different types of applications. We've looked at over 10 species, and you can see here just from a subsampling of the papers that are out there, they cover a wide range of topics, but the models have been extensively validated by both academia, NGOs, and then also, of course, the pharmaceutical and biotech industry. And it's also been extensively compared, which I'll get to in a moment, other types of 2D and spheroid systems that are in the marketplace today for the applications that I've described here. The products themselves, there's 10 different species, and they're shipped in a flexible format. So if we want to ship them with multiple species on a single plate, we refer to that as a zoo plate, and that's something we're able to easily do. And then when the plates arrive, after about a 4-hour exchange period, and a media exchange, we're able to use them right away for our assays. But the process is very flexible. They're shipped live right to your lab, and you can use them in your assays right away. And like I mentioned before, there's a 4-week, greater than 4-week stability of these, which is really just fantastic compared to most other models that are out there. And of course, fantastic compared to traditional 2D cell culture models that have been used for years, specifically primary human hepatocytes, which has been the go-to. We're able to get longer metabolic and phenotypic stability with the patented Hurel culturing process. If we look at the landscape and compare the Hurel models to micropattern models, 3D spheroids, what we see is that with the Hurel models, we have far higher number of hepatocytes per well. What this means is we have much better phenotypic stability, and we're much more easily able to see low clearance compounds and what happens to them, how they're metabolized, compared to other models where we have 1,000 or 5,000 hepatocytes. This system can go from anywhere 12 to 384 wells. And one of the unique things about the Hurel microlivers, which I'll get to later, is that we can actually look at hepatitis infection, and it sustains it for long periods of time. It's also compatible with seahorse, and for high content imaging, it's compatible with traditional high content imaging approaches. So it's very extensible to a lot of different applications, of course, DMPK and DILI, but overall, the greater number of hepatocytes and our unique culturing process allows this to be the most phenotypically and metabolically stable type of liver model in the marketplace. Like I mentioned previously, the Hurel microliver models have a very high degree of metabolic competency. And we see here a superior ability to generate reactive metabolites, as exemplified by Hurel's viability after exposure to hepatotoxin cyclophosphamide, incubated in the absence and presence, respectively, of broad cytokine P450 inhibitor aminobenzotriazole. And you see that on the right here in this graph, which allows for this high degree of sensitivity and also the predictive capability of the model itself. And then I mentioned this as well previously, but the phenotypic stability of the model has been peer-reviewed, and we see this for over 30 days. It's immutable compared to other models that are out there, that we have the highest degree of phenotypic stability in the Hurel model platform. And this is especially compared to hepatocytes in suspension, in monoculture, in micropattern arrays, and also in spheroids. One of the unique things about the Hurel microliver platform is that when we expose, for example, the human microliver model to midazolam in the presence of CYP3A4 inducer rifampin and CYP3A4 potent inhibitor kinaconazole, which we administer every 24 hours for 72 hours, and we remove these, what we see is the microlivers demonstrate a return to basal enzyme activity in a time-dependent manner, which illustrates the dynamic responsiveness and resilience of the Hurel microliver model metabolic competency, which is just incredible. It returns their basal state after we remove these, which really demonstrates the metabolic competency of these models and also their resistance. And when we look at this model for die-lie prediction, and we look at its sensitivity and specificity as compared to 3D spheroid models and micro-patterned co-culture models, all primary human hepatocytes, what we see is the accuracy and also the specificity and sensitivity of the Hurel microliver models is significantly higher, as you see here, based on the publications and literature out there in the research space, which is really incredible to see and shows another application of the model. And then when we look at metabolite generation, what we see is based upon a study from a major US pharma company, we show that the predictive capability of the Hurel microliver models is far improved compared to suspension, 2D cell culture, and other approaches that have been traditionally used, which demonstrates, again, the ability for this model to replicate what's happening in vitro compared to known in vivo outcomes. After going through the features of the product itself and some applications and data, I want to touch upon the services that we offer as a company. Visicol operates primarily as a contract research services company, in addition to selling the Hurel products as well as other reagents and kits. And what researchers are able to do is send compounds along to Visicol that we can test in Hurel microliver plates. We have LC-MS capabilities for metabolite generation and ID studies. And the Hurel microliver co-culture system is robust and reliable for human in vivo clearance studies, especially slowly metabolized drugs where we can't see the clearance in other types of models, especially suspension primary human hepatocyte models. The next application that we routinely do is Hurel Tox. This is a simple tox assay that we do, leveraging the system in various types of readouts. We can do imaging-based readouts, we can do ATP assays, we can look at albumin. There's various endpoints that we can do as a company, but again, what's happening here is clients are sending along to us compounds, and we're running those through simple and traditional tox assays within the Hurel system. We can go from as simple as ATP assays all the way up to looking at high content confocal imaging if needed at high resolution, and everything in between, qPCR, RNA-seq, ELISAs. We really have a wide array of different endpoints that we can look at, but it depends upon what the research question is. And we can also add a mechanistic readout to it as well to look at reactive metabolites and cholestatic liability as well. This is a very flexible platform, but essentially we're taking in compounds and testing in the Hurel plates. And then lastly, for services applications, like I mentioned previously, the Hurel microliver model is very stable with hepatitis infection. We can keep infection for long periods of time within the model, and of course subsequently test therapeutics against hepatitis, which makes it a really robust in vitro platform for this compared to other systems that are out there. And there's several publications like the Nature Communications publication you see here, reference that you can pull from looking at hepatitis in the Hurel microliver model for various mammalian species being used. And then lastly, for working with the Hurel microliver services and products, these are your main points of contact. The Hurel team has joined the Visicol team and will be part of the Visicol team going forward. So if you have any questions on the models themselves, ordering the models, setting up your next study, please reach out to the contacts here. And if you have any questions, don't hesitate to reach out. Thank you.